So our final speaker of the evening, thank you everyone for staying around, is Zoe June Assaf. Assaf? Yes. I've almost got it? Okay. And she will be speaking about mutational patterns in Drosophila melanogaster.
Cool. Thanks for having me here today. Okay, let's go. So mutations are the driver of all genetic diversity, providing the raw material upon which both natural selection and stochastic forces act. This means that if we go out and sample a genome from a natural population, what we actually find is just a subset of that initial mutational input. So for example, if we were to look at coding mutations, we expect naively that 75% of them will be non-synonymous. That's our neutral expectation. But if you get a sample of either all polymorphisms or common polymorphisms, you find that it's closer to 20 to 25% that are non-synonymous, showing that selection has removed the majority of them. However, one of the analyses we do of diversity either within or between populations will depend fundamentally on our assumptions about that initial mutational input. So how can we get at that? Ideally, what we would like to do is to get a set of mutations that are new before selection or drift have affected them. And so I'm going to discuss two different methods to do that. One is to experimentally generate them, and two is to try to identify them in natural populations. So I'm going to first briefly go through the experimental strategy. So a mutation accumulation experiment is a method to capture a set of new mutations by effectively keeping the population so small that selection's not very good at removing them. So what you can do is, for example, in this case, keep your population to size 2, and then take those flies through many generations such that any new mutations that occur can't be filtered out by selection. So you can either do this homozygously, so that new mutations are homozygous in the lines, or you can do it heterozygously. The advantage to this being that then recessive lethals or deleterious mutations can also accumulate. So far, I'm going to talk about 3 different experiments that have genome sequencing from MA lines. And there was another paper that came out last month, but that's not included in this analysis. So through these 3 experiments from 2009, 2013, and then our additional data, we have a total of about 700 mutations. And the main difference between them is that the mutation rate we found in our data is significantly higher than what people found before. We think this is due primarily to our different experimental design. So we did the accumulation in heterozygous lines instead of homozygous, so this allows recessive lethals to come through. Additionally, we did more lines for few generations, whereas the previous studies, when sequencing was more expensive, would do a few lines for hundreds of generations, which we think might have inadvertently selected for lines that have fewer mutations on average, since MA lines do die out. Besides this, these 3 experiments are very similar. So if you look at this, if you classify mutations into the 6 types, you find that the distribution or the relative rates of the 6 of them is the same across the 3 experiments. And from here on out, in other ways too, these 3 experiments are very similar. And so from here on out, I'm going to combine them so that we can have a higher count, basically. So do these experimentally generated mutations reach neutrality? And the answer is yes. So we expect, for example, that the non-synonymous fraction in coding regions will be 75%. And indeed, if you combine the 3 MA experiments, you find that you do reach the 75% expectation. So now can we look at other more subtle mutational biases? For example, context dependency. So is the mutation spectrum at a C affected by its neighbors, or the triplet context? We like to think of this in terms of the mutation spectrum at that C nucleotide rather than just the mutation rate. This will correct for any inadvertent biases to sequencing or mapping in the genome. So for example, if I look at all the different possible triplets centered at a C base, you expect a relative rate of C to T, C to A, and C to G to be the most to the least common. If we look at our MA data, you find that it's kind of hard to distinguish whether there's any variation in this mutation spectrum across the triplet context. And so overall, the MA experiments are really the gold standard for identifying the actual mutation rate. But our ability to detect any more subtle mutational biases is limited by the low count. So now I'm going to move on to the main part of my talk, which is looking for new mutations in natural populations. And what I mean by new mutations is really rare mutations. And that's because we can do that because rare mutations are on average younger. And additionally, because they're rare, their frequency dynamics are dominated by stochastic rather than selective forces. And so the spectrum of mutations we can see will be relatively unaffected by selection compared to common variants. So this is perhaps obviously complicated by the fact that it's going to be hard to distinguish sequencing error from a rare variant. However, I'm going to talk about how we can leverage 3 sets of publicly available data that's been made available to us from the Drosophila community, which is really, really awesome. So the first resource I'm going to talk about is the Drosophila Genome Nexus, which is really a compilation of many resources, altogether representing 621 individuals sequenced to high coverage. So these are, for the most part, either inbred strains or haploid embryos, and so the variant calls are made in a homozygous state. In this data set, we have high confidence in a given variant. However, its frequency isn't very high because very low, because we only have 621 individuals. The other data set we'll leverage is data that's come out of ours and collaborating labs, which is pooled sequencing of wildflies. In this data set, we have over 4,000 chromosomes represented from over 30 populations. And here we have a much larger population size. So theoretically, you can get variants at a much lower frequency. However, our confidence in a given rare variant is lower because each chromosome isn't that well represented in the sequence data. And then the third piece of data that we'll use is that there's been a number of DGRP lines that were actually resequenced. And so there's about 30 of these lines that were sequenced independently twice. So we're going to also use this data set. So how will we do that? What we're going to do is actually identify the variants that we're going to use in the DGN. So these are variants that we have high confidence in. So for example, if you find 1 copy of a variant out of the 621 individuals, it's a singleton. If you find 2, it's a doubleton. What we can then do to get at more rare variants, or variants that are more rare, is to look at those singletons and ask if they're present in the pooled data. If they're not present in the pooled data, then we can say that the frequency of that variant is at least 1 over 5,000. Even though we're pretty confident that these rare variants are real because they were identified in these high coverage genomes, you might still worry that this filtering down process could inadvertently select for sequencing errors. So to deal with that, we're going to also look at the resequence data to actually confirm these variants. This turned out to be very important because the rare variants were actually confirmed at a lower rate. So if we look at the variants at frequency 1 over 5,000, you find that where we started with 1 million variants, of which 15,000 were resequenced, we only actually were able to confirm close to 9,000 of them, or about 60%. This confirmation rate's much better in variants that are more common, as you might expect. So you find that, for example, doubletons have a confirmation of close to 90%, and common variants have a confirmation rate of close to 100%. So we wanted to know what was driving this because we didn't want our rare variants to be primarily errors. And so it turns out actually to be primarily due to regions that were difficult to genotype in the resequencing data. So if we look at our variants and ask what was the rate at which they were genotyped, you'll find that where common variants are genotyped in the resequence data at close to 100%, this rate goes dramatically down as you go to rare and rare variant classes. So what happens if you look at the confirmation rate within sites that you do have genotype data for? Then the confirmation rate is close to 100%. So if you look at the resequence data and just look at sites that have a call at all, our confirmation rate's very good. So we're not really disconfirming, we're just unable to confirm them. We think this is driven by difficult to genotype regions. So, for example, if you look at our variants at frequency 1 over 5,000 by location in the genome, you find that the genotyping rate is higher in coding and UTR regions than it is in intergenic, intronic, and promoter regions. This effect is mitigated as you go to more common variants. And however, if you look at the confirmation rate, it's within genotyped sites, you find it's robust to the location in the genome. One of the things we thought could be driving this was indels. So in our DGN variants, we actually do mask indels that are called within a given genome. However, we thought that perhaps we could get more information if we looked at each site and asked how many indels were segregating in other individuals. So to do that, we classified our rare variant, H variant class, so for example, 1 over 5,000, by the number of indels that were segregating in other individuals, and then asked what was the genotyping rate. If you do that, you find that with increasing indel frequency, as measured by indels called in other individuals, the confirmation rate at a given site decreases. So this suggests that our failure to genotype in the resequence data could be driven by the fact that there's an indel present that was incorrectly called as a variant, perhaps in the original data. This effect is mitigated again as you go to more common variants. And then if you ask what's the confirmation rate at sites for which we actually have genotype data in the resequence set, you find that it's close, again, to 100%. So I hope I've convinced you that it's important to actually resequence our rare variants, because it is possible that they could be errors. So I'm going to move forward with with the variant classes that have been confirmed. So I'll indicate that with a little c following the frequency. So when you do that, we have close to 9,000 variants at 1 over 5,000 frequency. And this is effectively an order of magnitude higher than what we had for the MA lines. So does this reach our neutral expectation? And the answer is it does approach it. So if you look at the MA data, you'll recall that that they reach the 75% non-synonymous fraction we expect within coding regions. And the common ones are, again, close to 20 or 25%. But then you can see as you decrease your frequency, you get closer to that neutral expectation, where for the variance at 1 over 5,000 frequency, you reach 68%. Interestingly, if you look at nonsense variants, you don't really see this. So you expect naively that 4% of coding mutations will be nonsense. Whereas in the MA lines, we weren't really able to see if we reached the neutral expectation because the counts were so low. And in common variants, you very rarely see nonsense variants. You find that in even the most rare frequency class, you only ever see 1% of mutations being nonsense, which suggests we're actually missing 3/4 of the expected nonsense variants. Another nice sanity check is to look at germline expression. So you would expect that if you were to look at common variants present in highly expressed genes, that you might find fewer of them in highly expressed genes, because the idea being that because they're highly expressed, they're more important and so less tolerant of new mutations. And so you can see that effect in the rightmost graph. When we then look at our more rare set, you can see that that effect no longer exists, suggesting that we're capturing these variants kind of before selections had a chance to remove them. So now let's return to our question of context-dependent mutation spectrum. Can we actually measure this? So if you'll recall, we're interested in whether the spectrum of C mutations, C to T, C to A, C to G, changes based on triplet context. And you'll see that now we do have the count to be able to start measuring triplet biases. So for example, you can find that CGA, GGA, and TCA triplet contexts have a higher C to T mutation rate compared to the other two types of C mutations. So in conclusion, our new MA data suggests a higher rate in Drosophila melanogaster than previously found. And additionally, because of the availability of a number of awesome resources from the Drosophila community, we're able to detect a nice class of very rare variants and use those to measure other aspects of the mutational spectrum. So I'd like to thank my advisor, Dmitry Petrov, who's been awesome, as well as Suzanne Tilk and Jane Park, who helped with the MA work, my funding, and the rest of the amazing Petrov lab. And thanks. I'd like to take questions. Or everyone just wants to go get a beer. That's cool, too.
How was the mutation accumulation experiment done? Against a balancer or a marked strain?
We used a marked stocked that we repeatedly backcrossed to, so that you would always take, kind of similar to what we saw before, where you end up with red eyes, brown eyes, or white eyes, and you take the red eyes every generation.
Do you kind of look for gene conversions, like, and know that they aren't happening?
Could you say that
again? Do you
again?
kind of look for gene conversions and make sure that's not like something's not coming off the mark strain into the...
Yeah, we didn't check for that, but that might be a good idea. Cool.
Does the fact that you're missing so many nonsense mutations, do you think that's because around 3/4 nonsense mutations are lethal? Is that what
Or
you think
strongly
is going on?
deleterious? Yeah.
Okay.
Yeah, we're working on seeing if we can get an actual likely minimum selective coefficient for it.
Okay, I think that wraps up our session. We'll see you again tomorrow morning, I hope, and let's thank all of our excellent speakers.